Hello students, in this lecture video, I'll be discussing the concept of meiosis. Now, meiosis basically occurs during the sexual reproduction when gametogenesis has to take place. Now, whenever gametes are being formed in plants, the process of cell division, that is meiosis, it takes place. Now, in meiosis, there are two phases. One is meiosis 1 and another is meiosis 2. But there is only single cycle of DNA replication. Now, let us look at that how in two phases what is happening. So, after S phase, there is meiosis 1 and then meiosis involves the pairing of homologous chromosomes. Now, when homologous chromosomes are paired, then there is meiosis 2 and after meiosis 2, 4 haploid cells will be formed. Now, the meiotic divisions, they are divided into two phases, means in meiosis 1 and 2, there is karyokinesis and cytokinesis. So, the phases remain the same, only the events change, what the events are happening. So, in meiosis 1, it is prophase 1, metaphase 1, anaphase 1 and telophase 1. And meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2. So, let us look at it that how in meiosis 1, what are the different phases and what exactly is happening in meiosis 1. Now, in meiosis 1, that is also divided further into 5 phases. Okay. Now, what are those 5 phases? Leptotene, zygotene, pachytene, diplotene and dikinesis. So, these are the phases, 5 phases where the chromosomal behavior will change in meiosis 1. So, in leptotene, that is the first stage, chromosomes start appearing, means like in nucleus there is a chromatin thread, it will start forming compact structures called as chromosomes. Then in the second phase, that is zygotene, the pairing of homologous chromosomes will start, that is they will start pairing like this. After this, you can say that when they start pairing, there is a protein complex which enhances the pairing process. That is called as synaptonemal complex. And at this phase, you can say there are 2 chromosomes or 4 chromatids. 1, 2, 3 and 4. That is why this stage is called as tetrad. And because 2 chromosomes are there, so it is also called as bivalent. Next is pachytene, wherein what is going to happen, this stage is characterized by formation of recombination nodules, where the exchange of genetic material will occur. Finally, there is a recombinase enzyme which will help in this process. It will also help in crossing over, which is enzyme mediated. Finally, diplotene, where what will happen? The homologous chromosomes, they will start separating, but at some point they will be attached. Those are called as crossovers. And this region is also known as chiasmata. Finally, there will be separation of these chromosomes and meiotic spindle will start forming. Again, the formation of spindle fibers. That is called as dikinesis, that process. Now, after this prophase 1, which is for longer duration, there is metaphase 1. Now, in metaphase 1, what is going to happen? These chromosomes, they will align at equatorial plates, similar to metaphase of meiosis. Then, coming to the different next stage, that is anaphase 1, where the homologous chromosomes, they will separate and the sister chromatids, they remain attached to the centromere. That is the difference. Here, splitting of centromere will not happen. You can see here, okay, in anaphase 1. Then in telophase 1, there will be nuclear division where you can say 2 cells are formed. So, it can be called as dyad of cells. Now, this stage when there is, you can say, nuclear division and meiosis 2 has to begin, that in between stage is called as interkinesis, which is followed by prophase 2 and it is very short-lived stage. Now, what is happening in meiosis 2? The prophase 2, meiosis 2 is initiated after cytokinesis and again the nuclear membrane will disappear and chromosomes will become compact. Similarly, at metaphase 2, there will be alignment at the equator. At anaphase 2, the splitting of centromere will start and they will move towards opposite poles. You can see here very well in this diagram that how they will move to the opposite poles after splitting of centromeres. And in telophase 2, like the mitosis, it will again get the nuclear envelope and you can see 4 daughter cells. So now these are haploid. Finally, let us look at significance of meiosis. Firstly, it helps to conserve the specific chromosome number, and then it results in reduction of chromosomes to half. Because of crossing over, genetic variability is seen, which will lead to evolution.